All right, guys, so now we're continuing. So we now have a table of smoking results. So we've compared our two groups. And now let's load up a library that allows us to annotate those, because right now we have, let's look over here. We basically have a group of uninformative probe names, and we want to know what genes those probes actually map to. And so let's load an annotation library that allows us to annotate that. And this code chunk here basically says, okay, create an ID column in our smoking results and create a symbol column in our smoking results. And when we enter that in, we now get gene names alongside these probe names. And then now, I mean, the analysis is done, the comparison is done. We have statistically significant differences between our two groups, smoking and non-smoking, and now it's all about getting in and exploring that data. So we have this long code chunk here, and first we load a library called ggplot2, which is a very popular library that is designed for data visualization. And so we're not going to get into everything that this particular code chunk means. I'll provide it to you, so I'll upload my R notebook of these commands for you to enter in, make it a little easier so you don't have to type all this out. But basically, we're now trying to identify specific trends within our data. And so within our code chunk here, we've set specific thresholds to actually highlight our particular point. So each point here is a particular probe that we tested for gene expression changes between our smokers and non-smokers. And we said anything that's below 0.05 adjusted p-value, label in red for us. And then we can also do things like take our different genes and actually identify them based on specific identifying numbers, so that we can see the specific genes that are most down and upregulated in our data set. And then we can even look specifically at individual genes. And so this is MERK, which you'll recognize from the title of our journal article. So this was what was found to be upregulated in smokers' macrophage cells as compared to non-smokers. And we can actually look specifically at that gene of interest, and you can see that in smokers, the gene expression is upregulated compared to non-smokers. And then we can also start to make what's called a heat map, which basically has our sample in the columns and our genes, or in this case, these specific probes as our rows. And basically it colors it by gene expression intensity. So yellow in this case, if we look at our legend, being more upregulated. And so these at the top are more upregulated in smokers, compared to these down below are by and large downregulated in smokers. And then we can start to play around with colors. Whatever we think best represents our data, we can put smoker in red, non-smoker in blue, and do specific things like that. And with that, that's kind of our first very quick exploration of a gene expression data set. So please, when you're finished with these commands, go ahead and, as you went to file and created a new file and created the R notebook, and entered in your commands as we moved along. When you're done with that, go up to knit and knit to HTML. And once you click that, it'll actually run through them, and it'll produce a data file in your working directory. That's this so I called it microarrayactivity.html, and you'll be able to open that in any browser, and this will actually walk you through exactly what commands you entered and the output of those commands. And so you see that we're reading in, we produce our box plot, we normalize, we have our normalized values, and then we do our specific comparisons. And so this is what I want you to turn in when you're finished running through these commands. And for the rest of this week, we're gonna kind of move into kind of the cutting edge of RNA-seq, which is this idea of single cell RNA-seq. And so instead of actually looking by sample, we now have the technology to look at the gene expression in one particular cell, and we could do that in thousands of cells at a time that we're going to see in the next activity, also using R. And with that, thanks for listening to this online walkthrough of R and exploring this gene expression data set. Stay healthy and stay safe, guys.